These small darting finches, Darwin's finches, are found only in the Galapagos Islands. They've become symbols of the most revolutionary idea in biology, that life evolves by selecting from the rich diversity of nature. From 1831, Charles Darwin spent 5 years on board the Beagle. Circumnavigating the globe, he followed the best of Royal Navy traditions. Earlier voyagers had discovered remote islands strewn all over the Pacific, Indian and Atlantic Oceans. During his visit to these islands, Darwin filled his notebooks with what he saw and collected species without recognizing any pattern to the life of oceanic isolation. The full impact of his observations came much later, when he correctly interpreted the flora and fauna of each island as species in the making. They had evolved. And he proposed that successful species had adapted to their environments by a mechanism he called natural selection. Whether natural selection can be the only driving force of evolution is being seriously debated today, in the light of remarkable discoveries being made at the molecular level. DNA, the genetic thread that links generations, is also a significant source of diversity. We now know that changes that arise by chance in DNA are passed on to the working molecules of life, like hemoglobin, which carries oxygen round in our blood. Some suggest that such molecules continue changing over evolutionary time, oblivious of natural selection. This idea that molecular evolution is neutral has split biologists into two camps. According to Darwin, some of the best evidence for natural selection comes from the oceanic islands. Our port of call? Madeira, a tiny archipelago 100 miles off the west coast of Africa. These islands attracted the great 19th century geologist Charles Lyell, who proposed a theory that the Earth evolved. He thought that such islands surfaced from volcanic activity deep in the ocean. Lyell's theory was one of gradual changes, and it was a major influence on Darwin, who came to believe that life, like the Earth itself, evolves. On Madeira, we find the geological evidence that gave birth to the island, but what of life? Born by wind and ocean from Europe and Africa, came the ancestral species so crucial to Darwin's theory. Today, geologists can date the rocks of the islands precisely. They're just 12 million years old, a relatively short timescale for life to establish itself here. In The Origin of Species, Darwin wrote,
"Species arriving in a new and isolated district will be eminently liable to modification and will often produce groups of modified descendants. We see that this is true in the endemic birds of the Galapagos Islands or the endemic land shells in Madeira."
Lawrence Cook from the University of Manchester has found some fossil remains of early arrivals. Do the Madeiran land shells verify Darwin's important idea of modification producing groups of modified descendants? The fossils can be compared with modern descendants. There are resemblances, but also differences. Evolution has taken place. And over 200 groups of modified descendants, separate species, have now been recorded, many of the snails being unique to Madeira. In adapting to new environments, any inherited differences that increase the chances of survival and reproduction are passed on to the next generation. Natural selection at work. The snails in this area fit Darwin's scheme. They live inland, away from the heat, and under cover in the damp forest conditions. Dark species are favoured in these shady conditions. They look very different from the almost white species living in sunny coastal climates. Yet these snails share the same ancestry. But natural selection needs variety to select from. And we've discovered that pool of diversity in the genes. These snails, Sapir nemoralis, are all one species, and they express a wealth of genetic diversity on their shells. Steve Jones.
People have asked the question, why is that diversity there? Is it acted on by natural selection at all? And an obvious way to approach that is to look at populations living in very different environments and seeing whether they have different patterns of genetic diversity. We chose to work in the Pyrenees because there you have extremely abundant snail populations living in very different places, right the way from about 2,500 meters above sea level down to sea level in habitats which differ from alpine tundra right the way down to sand dunes, obviously under very different climatic and ecological stresses. And what we did was to map out patterns of gene frequency.
The experiment covered a vast area of 400 square kilometers in the central region of the Pyrenees. It centered on a cluster of river valleys surrounded by mountains rising up to 2,000 meters high. What was the plan for the fieldwork? They collected a sufficiently large number of snail populations at They counted the frequencies of two genetically different types of shell, those with bands and those without. At the top of the slope, there was a high proportion of light-coloured, unbanded snails. But the darker, banded snails predominated at the bottom of the slopes. Many aspects of the environment change with height, but there's an interesting feature in the climate. At night, cold, heavy air flows down the mountain slopes and accumulates in the valleys, so the morning temperatures may be much lower than those on the high slopes. Just as our own comfort on hot summer days is helped by white clothing, snails with genes for light shells will be favoured in places where there's most heat, at the top of the slopes. Genes for darker shells will be favoured in colder areas at the bottom of the slopes. This pattern to the diversity held for dozens of valleys in the region. So this extensive undertaking, where thousands of snails were sampled, shows that climate causes genetic changes between snail populations in the Pyrenees. Sapir nemoralis also lives in Britain, and the Marlborough Downs is Steve Jones and his colleagues want to discover how genetically different individuals sampled from diverse populations are acted on by natural selection. The snails in this part of the Downs are polymorphic, genetically different individuals that can be identified by the patterns of their shells. They're just what Steve Jones needs for the experiment he has in mind. He plans to monitor the individual behaviour of 5,000 snails in an effort to understand the link between single animals and their environment. Just as in the Pyrenees, on the Downs, natural selection by climate does produce genetic differences between snail populations. In sunny, warm, dry places, there are more light-coloured snails. But studying the genetics of individual snails requires an experimental control that's usually only found in the laboratory. To do the experiment in the field, Steve Jones takes his experimental animals to their home for the summer. He's built what he calls a snail ranch on the edge of Whiteham Wood near Oxford. To measure the influence of climate, he simply dabs each snail with a spot of paint that fades with exposure to the sun. By comparing two genetically different individuals from the same population, some with effects of the snail's genes on their behavior. He tests for differences that may be directly related to their shell pattern by painting the dark shells white and the light shells black. Once all the snails are collected, painted and in residence, the experiment gets underway.
As far as we know, this is the only snail ranch in the world. And you might ask why we've put so much effort into farming mollusks. Well, the answer is that this is, in some ways, a halfway house between the field and the laboratory. Our climatic sensors allow us simply to put the snails in our experimental cages in April and come back in October and ask how much solar energy have single animals experienced, and is that related in any way to their genetic constitution? What we'd like to do is to get an idea of the sort of differences in the environment, in climate in particular, which single animals might experience over a whole year. So it's a sort of whole season experiment.
Autumn marks the end of the season, and it's time to recapture the snails. Over the summer, individuals have been living in different amounts of sunshine. Some have spent their time on top of logs, but most are hidden under the dry grass and nettles. And some snails have buried themselves completely. Different patterns of behavior, but are these under genetic control? And how does the green paint measure these differences? Back in the lab, the painted snails can be scored accurately against a standard scale of paint fading. The lighter and more faded the spot, the more exposed the animal has been to the sun. Histograms of fading scores can be built up and interpreted. On average, paint spots on banded snails are more faded than those on the unbanded ones. So it seems that banded snails are exposed more to the sun than the unbanded ones. In the same way, black snails are shifted in the direction of more fading and the white shells of less. So single snails from a variable population do respond differently to climate. Their behavior is related to their genetic makeup. Is this linked in any way to the action of natural selection?
We know that snails, in common with many other animals which were perhaps rather mistakenly known as cold-blooded, get the great majority of their energy not from their food, but directly from sunlight. what we found is that this difference in thermal relations in sunshine has a major effect on their behavior, because what we've now got is a system where we have a genetic polymorphism at the genetic level, which alters the appearance, the phenotype of the shells. It makes them darker or lighter. That in its turn, for simple physical reasons, alters the extent to which an individual obtains energy from sunlight, and that is also manifest in the single animal's behavior and ecological niche over the period of a whole season.
So we can begin to devise a mechanism that links genes to physiology, to behavior, and most important, to natural selection. It's just the mechanism that Darwin predicted. But he also knew that natural selection was likely to be a very complex process.
Studying genetics of snails is a beautiful example of how to be a successful scientist. That is, to know where to stop. Because the more we learn about polymorphism in Sapir, the more well it is illustrated that nature is really pure and never simple, because there are lots and lots of different kinds of evolutionary forces that seem to act on this shell polymorphism. And a very important one, at least in some places, is natural selection by predators.
A common predator of Sapir is the song thrush. In spring, when the birds are nesting, they often hunt for snails on the ground. In their search for prey, they recognize the common shell patterns in a habitat. This feeding behavior is rarely seen. But these stone anvils and the debris that surrounds them are commonly found on the downs. Since the birds tend to overlook the snails carrying genes for rare shell patterns, a polymorphic population will be maintained. This frequency-dependent selection could be important in maintaining visible genetic diversity in the wild. Can natural selection act on the huge amounts of hidden diversity that we now know exists at the molecular level? Less than 30 years ago, a remarkably simple technique called gel electrophoresis revealed a wealth of molecular variation. This measures the speed at which molecules, proteins in this case, travel in an electric field. As each protein variant has its own characteristic speed, the variation in these tracks reveals that most proteins are polymorphic.
In many ways, the discovery of things like protein polymorphism, which are gigantic mountains of diversity in some ways, was an embarrassment for population genetics. Because it's very hard to see how natural selection, the creative force of evolution, can act on all this genetic polymorphism, simply because it would be so expensive. If it was the case that every single enzyme variant conferred some advantage or disadvantage upon its carriers, then it would be enormously expensive to try and maintain this variation by selection, because inevitably some individuals, a very high proportion of the population, would have to carry variants which made them disadvantageous and might cause them to die or to fail to pass on their genes. And this is the genetic load, as it's sometimes known, the cost of maintaining variation by natural selection.
What are the patterns of molecular variation in the Pyrenean snails? The differences are revealed by gel electrophoresis in an analysis of the enzymes of snails sampled from the mountain slopes. The variation for each enzyme shows up as tracks of different lengths on the gel, because in this analysis, the speed of tracing the bands will differ for each variant. When any two variants for an enzyme were scored against their sampled positions on the mountain, there was no association with height at all. In fact, there was no association with any environmental change, such as climate or predators, as already established for shell patterns.
Therefore, almost by default, we're forced back to the position that this may represent some other kind of evolutionary force, in fact, evolution by accident, by genetic drift.
Genetic drift has been the main area of research for Japan's leading population geneticist, Motoo Kimura. It was variations in proteins like hemoglobin, particularly in fish, that led him to controversial ideas about how evolution works at the molecular level through changes in the DNA itself. Here's how it works. The language that codes for protein molecules is spelt out in a simple chemical alphabet, the 4 bases in the DNA molecule. The code itself uses triplets of bases, and DNA makes proteins through messenger RNA. RNA has its own sequence of triplets complementing those of DNA. The RNA message specifies the different amino acids that are the building blocks of proteins. There are 20 distinct amino acids coded for by the triplets, and the combinations of these amino acids make up proteins. Let's look at some examples. GUA codes for the amino acid valine, but so do other triplets. GUU, GUG, and GUC. In this case, the third letter of the triplet makes no difference to the amino acid that's made. So from the point of view of evolution, it's a silent change. But other alterations can make a dramatic difference. A change in the first letter of the triplet from GUC to UUC spells a new amino acid, phenylalanine. And a change in the second letter, say from UUC to UGC, spells cysteine. Natural selection is more likely to act on the first and second letter changes, whereas the third letter changes are silent, unaffected by natural selection. Kimura first used gel data and later the new information on triplet changes to calculate rates of evolution. To his own surprise, they were much higher, more frequent than expected if evolution works by natural selection. For Kimura, these substitutions and their rates meant a new interpretation of evolution.
1960年の始まりでした
I applied the estimated rate for amino acid substitutions to the amount of DNA that could change over evolutionary time.
速度が求められるようになったわけです。でそれを
My calculations showed that nucleotide substitution in different species has been so fast that, on average, one DNA base pair has been altered in the population roughly every 2 years. We can only reconcile this high rate with the limit set by genetic load if we assume that most mutations involved are selectively neutral, or almost neutral. So, most of the changes we observe must be the result of neutral mutations being established by chance through random genetic drift.
The neutral theory of evolution, developed by Kimura and his colleague Dr. Ota, has had great success in accounting for molecular change. But still the controversy remains. The challenge to Darwin's theory is to explain these molecular changes in terms of natural selection, the mechanism that accounts so well for those variations in form that he first discovered over a century and a half ago.